So let's get started. So why is recombination important? We probably all know it, but just quickly remind you, most importantly, it can bring beneficial mutations together. So when two mutants, beneficial mutations arise in a population, with recombination, they can get to the same genetic background, and therefore the beneficial mutant big A, big B appears in a population sooner than if there is no recombination and the mutant big A, big B has to arise from the background of, in this case, big A, small b. So that's clearly a benefit of sexual reproduction, of recombination. Also, it maintains genetic diversity, purges deleterious mutations from the population, and as I said, in eukaryotes, is provided by sexual reproduction. Well, this is clear, but how about in bacteria? Well, clearly we know that bacteria reproduce by clonal reproduction, binary fission, yet they have ways of exchanging genetic information. It's been shown already in the 1990s, in 1993, that many bacterial species or their genomes are in so-called linkage equilibrium, which means that different variants of certain genes are not linked. Essentially, the genomes of different, let's say, isolates are freely recombining. There are no association between different alleles. Here is like a list of species where it's been discovered. And clearly one can think of the ways of horizontal gene transfer, and that competence, natural competence, would be a way of achieving this linkage equilibrium. But not all the species that are in this list can do can do natural transformation, are not naturally competent. So there must be some other way of doing recombination. Clearly, yeah, and also I wanted to point out that natural competence is or has been documented in only 80 bacterial species, so it's probably not that common. So the only remaining way of potential recombination is transduction. But how come it is so frequent? Because when we have a phage that's doing generalized transduction, it's usually a big threat for the bacteria around. So we think about I was thinking about ways how this can happen. So clearly, let me put it this way. My research question was whether can bacteria adapt faster by transduction-mediated recombination than by de novo mutation. Well, as I said, like normally the phage would kill all essentially the whole population. So if there was only the transducing phage and the bacteria, then not. But bacteria have evolved multiple immune system, and as I had in my title, one of those is the CRISPR system, which protects the resistant cells from being lysed by the phage. Also, we showed a couple of years ago that the CRISPR system can provide herd immunity. And that's important because herd immunity can enable coexistence of resistant and susceptible individuals and the phage in the population. So now I'm gonna briefly describe this concept of coexistence of resistant and susceptible individuals in the population, which then may lead to gene flow from susceptible to resistant cells and therefore continuous recombination. So here is what or how we described this bacterial herd immunity. The main quantity is the herd immunity threshold, which depends on the phage burst size and also on bacterial and growth rate and phage latent period. And why is it important? Yeah, this is the paper in eLife about if you want to learn more about herd immunity. And why is it important? It's simply because once the frequency of the herd immunity threshold is reached by the resistant strain, it cannot grow further. It cannot go to fixation. So here in the plot, it is depicted that the resistant mutant can grow to a certain quantity, but once it reaches this frequency of herd immunity threshold, it's not selected anymore because the pathogen stops spreading in the population. So it reaches certain equilibrium. I'm going to talk about it later. Okay, so we have the principle, we have the hypothesis that CRISPR immunity, which provides herd immunity, can lead to continuous gene flow and recombination in the population. But this is just a hypothesis, so I'm gonna show how we tested it. First, we engineered a system for detection of recombination. We engineered 2 strains which had a chromosomally encoded loci. The first strain had a GFP, which was controlled by the PRM promoter from the lambda phage, which was also a bit injuring, such that it would work better, and a spectromycin-resistant cassette. And we put this locus in the lambda attachment site of E. coli. And then we engineered a second strain, which had the C1 transcription factor, which activates the PRM promoter in another phage attachment site, the P21 attachment site, accompanied with kanamycin-resistant gene. So it went here. For simplicity, we can think about these 2 loci as a bulb and a switch. So one is has the bulb, which is the GFP, but it can only be expressed when the switch is present in the same genomic background. Okay, so when these 2 recombine in the same background, we see fluorescence of the cells or of the colonies. Okay, so that's the first component. And then, yeah, here you can see the difference between the recombined and the strain and the strain 1 with just with GFP. And then we engineered CRISPR immunity to the P1 bacteriophage. So we took CRISPR-Cas9 from Staphylococcus, engineered 2 spacers targeting the P1 bacteriophage, and also added mCherry such that we can distinguish resistant and susceptible cells. And the control plasmid in the susceptible cells had no immunity, just to Cas9, but not targeting anything, and no fluorescence. Right? And then here we can see that the fluorescent resistance plasmid is clearly distinguishable. And then we combine these 2 strains with the plasmids such that the resistant strain had the switch, the C1, and the kanamycin resistance in the chromosome. And the susceptible strain, which is not fluorescent, had spectromycin resistance and the GFP, the bulb, in the chromosome. Right. So this is the experimental system. And now what happens, like when the gene flow from the susceptible to resistant cells occurs. So when a phage infects the susceptible cells, it replicates, and with certain probability, it transduces the locus with the bulb, with the GFP and kanamycin genes. Then the cell lyses, and when the resistant cell, which is in the same population, gets infected, it effectively eliminates all the wild-type viruses, all the wild-type phages from the population, and the GFP locus, the bulb, can get recombined into the chromosome and starts being fluorescent. All right. So we wanted to test, like, to what extent this effect or hypothetical effect plays a role in bacterial populations or in their or in the within population dynamics of their evolution. And so we mixed the 2 strains in a ratio close to the herd immunity threshold, and we added a phage with a multiplicity of infection 0.1 and inoculated that in a tube. And then we propagated the tube every about 12 hours with 1:2 dilution, which accounts only or allows for only 1 generation on average per transfer. In these settings, there is initial growth for about 3 hours, and then the culture reaches stationary phase. And that's very important to say that there is no selection for kanamycin or spectramycin resistance in the medium. So there are no antibiotics apart from those that are maintaining the plasmid. Okay, and then we sampled regularly on 3 different types of plates. So we sampled on selective plates to count mutants and recombinants, and non-selective plates to count the bacterial concentration population, and also plaque assays to count the phage concentration in the tubes. Okay, so let's finally get to the results. Here we can see on the left side that the resistant and susceptible cells coexist in the population, or that the susceptible strain doesn't get wiped out. On the left here in green, we can see the experimental treatment, which had resistant cells, the susceptible cells, with the spectramycin resistance locus and the phage. And then I also did it with 2 controls, and it is the magenta and cyan on the right side. And in the magenta treatment, there is no spectramycin resistance and no GFP in the donor susceptible strain, but there is a phage. And in the green, it's another control. There is no phage. There are the same strains as in the experimental recombination treatment, no phage. Okay, so we can see that indeed the susceptible strain remains in the population. And here the arrows indicate where I did the 1 and 2 dilutions. Okay, second question, does the phage remain in the population? Yes, the phage does remain in the population. Here we can see again time on the x-axis and the dilutions at the time 12 and 24 hours. And here we can see that in both treatments where we add the phage, it converges to the concentration of about 2 times 10 to the 6 plaque forming units per ml. They differ in the concentration at the beginning. Unfortunately, I don't have time to talk about it, so we may discuss it after, like in the chat or later on. And finally, I'm getting to the most interesting slide of my presentation, which is showing like whether the bacteria recombine faster or adapt faster by recombination than by de novo mutations. And it is first I'm going to show you the controls. So again, in magenta, we have the resistant cells, the susceptible cells without the detection locus and spectramycin resistant genes with the phage. And then in cyan, we have resistant cells, susceptible cells with the spectramycin resistant and GFP, and with no phage. Here we can see that it is pretty much consistent with prediction of evolutionary theory that mutations are random, and since they are not selected for in the medium, they remain essentially constant. The concentration remains constant. But when we allow them to recombine, we have to zoom out, because if we have the treatment with resistant cells and the donor strain with the locus we can select for, we can see that the fraction of recombinants, which fluoresce in green, steadily grows throughout the experiment. From these data, we can calculate how frequently they recombine. And if we calculate the recombination rate per gene and generation, we get to 6.5 times 10 to the -7, which is not that high, but it's for this particular gene or locus, which we engineered. But in the whole population, This happens for all genes. So this is just a proxy for like the total recombination. And if we calculate it for the whole genome, we get to the number of about 3 × 10⁻³ per individual and generation, which means that every generation, 1 in about 300 cells, is a recombinant of certain part of the genome. This is all I wanted to tell you. Hope I made it in time. So to conclude, I can say that the CRISPR-based herd immunity enables ongoing transduction-mediated recombination in phage bacterial communities. This recombination occurs mostly in stationary phase, and that adaptation by this recombination outweighs adaptation by de novo mutation by 1 to 2 orders of magnitude. Finally, I would like to thank all the people and labs who allowed me to do this work. So first, it is Pavel Plevka and his lab at the CEITEC, Central European Institute of Technology in Brno, Czech Republic. Then it was Jonathan Bolbeck from the University of Liverpool, and Karin Guet, Matto Lagator, and Claudia Igler back in 2018 at the IST Austria.